So today I'll be talking about the drug design, discovery, as well as the development perspective. Okay. So when I asked about the audience, your HOD ma'am told that it is going to be the faculty members. So I proposed like two type of approaches where a good medicinal chemist can start their work in spite of being anywhere in India, instead of not giving an excuse that because as head of the department, I always receive like we don't have softwares, we don't have sophisticated instrument, we don't have hi-fi labs, so we are not able to do good medicinal chemistry work. No. So even at a small lab, when you have a small basic fuming hood, you have a rotavaporator, you have a heating mantle, a small amount of cost in the chemicals, consumables, you can do good work. So I'll be talking more on two aspects where medicinal chemistry needs to work more. So it's not like only drug design where we always work. We have to focus more on the drug development phase also. Okay. So I'll give a general brief like what are the various techniques from the old methods of drug design till the current methods, how they are used. Okay. Yeah. So I'll give you an overlay. What are the challenges where everyone is aware being a faculty? So I'll rush up my slides on the basic understanding. And then I'll focus more onto the metabolism guided drug design and the scaffold hopping. So these two approaches anywhere in any parts of India, wherever you don't have to be in a reputed national level institute or state university, any small group of institution anywhere in rural part also, you can do very good medicinal chemistry work, provided you have a keen interest, your attitude is good, you start reading more on the medicinal chemistry perspective. Okay. So the contents generally I'll be talking is the basics like roots of drug discovery, the modern drug discovery, the modern challenges. The first three titles I'll try to push fast, and then I'll speak more on scaffold hopping, then metabolism guided drug design, where these two have very good scope for medicinal chemists. Because we always think we need to design drug. So getting a molecule, isolated molecule in vitro assay is bit easy than to bring out a molecule at the market level, because the molecule fails at many of the ADME profiles, which needs to be addressed. And if time permits, I will talk a little bit on the artificial intelligence, what are the algorithms are being recently used. Okay. So we know, like, as I always mention, like drug design. So medicinal chemistry does not play a role only in drug design. He or she has got an important role in the drug development phase also. So getting a drug from a known scaffold or start from the de novo design where you have no idea about the scaffold, getting from the hundreds of molecules, getting a lead, getting a hit is good. But converting from the drug discovery to development phase, we always think that it is not our cup of tea, or it is to the formulation or other departments who is going to play a very important role. Now try to understand, when we identify a lead or a hit molecule, when it is an isolated in vitro assay, the molecule is going to be really very good, beautiful. It is going to give a very good potency at a micro piconanomolar concentration, no doubt. But when you put the molecule into a complex system, even if you go into in vivo study, the basic animal model, then we move to the complex human body. What the body does to the drug and what the drug does to the body, both the pharmacokinetics and pharmacodynamic parameters are very, very, very important. So there comes important parameters like ADME tox. Though we use Lipinski rule of 5, it's just a mere starting point. You know, like almost all the drugs in the market, antibiotics, not necessarily they have to pass through the Lipinski rule of 5. It's just a small, you know, like a torch when you look for a needle in a dark room. Okay. So you always keep in mind that before you design a drug, or when you design a drug, before getting into the in vitro activity, please look for ADME toxicity. Once it is good at that profile, then go for the synthesis and then the activity. Because 90% of the drug fails at the metabolism or the toxicological profile, or it is get excreted easily from the body, where the clearance is high, the half-life is very low, and it produces some toxic metabolite. Okay, so this talk will be morely on to the drug development phase. When the drug fails at the ADME profile, remember the molecule is having good in vitro activity. It has gone to the next stage. It has failed in the development phase. So from there, how do we go back to find the molecule, to rework on the molecules where we have lost? So we know this is a very old diagram. Drug discovery is always an interdisciplinary approach. I'm not going to talk much, where you always have minds from different disciplines. I mentioned only 3, but other departments are there: core science, genetic information, structural biology, molecular biology, synthetic chemistry, computational chemistry. So all it's a multidisciplinary approaches. So the process of R&D to reach from the individual isolated molecules to the market takes minimum of 12 to 14 years, and the cost is really on a higher side. So this is a basic layout what we talk about, from basic research to new drug application filing, target identification, lead, candidate, preclinical development, filing, investigation, new drug, clinical development. So once we identify, we do a basic research, identify here. Now we'll be focusing only on the molecules where it fails in the pharmacokinetic parameters, where it shows some toxicity. Based on that, we'll try to do the chemical manipulation at these 2 points. So this is a graphical representation or pictorial representation how the modern drug discovery works. We know that now with the advancement of gene, proteomics, genomics, structural biology, we'll be able to identify the gene where is a problem. Then the important thing is, once you identify the target, you have to validate the target. There was a lot of drugs, including COX-2, which was successful or valid target around 25-30 years back, now faces a problem with cardiovascular disorder and diseases. So the target what you choose should be Pakka validated. So in the sense that it should not involved in any other metabolic pathway. It should be a proper target for that particular disease, conditions, or disorder. Once you get this target validation from the biologists, then you start working as a medicinal chemistry from the literature, or you try to develop a lot of lead molecules, read profiling, and then try to optimize, and then give it to the pharmacologists, where they start with the preclinical, clinical. So if it is good, then we are happy. And if it is bad, again, try to understand what is the problem with the gene level. Where is the biochemical pathways interfere? What is the enzyme? Where is the cascade is troubling? Everything. So this, in short, is target selection, hit identification, candidate identification. So from the clinical data, trials, that means from bedside to the benchtop. So you'll get some information at the preclinical or clinical sides when you're doing at the human level. You try to find some issues or problems, that is from the bedside, from phase 1 or phase 2. Then you there itself you try to address the problem, what is the metabolite, what is the reason, and then you directly go to the bench work, and again you have to start. That is what you call it as the translational research. So in those days, it was like, I always suggest that it was the medicinal chemist, where you do hundreds of molecules randomly, like combinatorial library I'll be talking about. So you produce hundreds of molecules. It is randomly screened by high throughput screening. And then after thousands of molecules, you come to hundreds of molecules as a hit molecule. Now it is completely vice versa. We know the target, we know the protein, we know the disease condition, we know which to block, which to inhibit, so that the undesired biological response can be stopped. So from biology, we try to identify what is the mechanism involved in the biology. From there, we go back to the chemistry. So the 30-40 years back, it was chemistry driven towards the biology. Now from biology, we move on to the chemistry, where you exactly know what is the lock. We try to find a key to open the lock and key, or a receptor inhibitor type of mechanisms. So these are nothing but the general methods. Like 50 years ago, it was very difficult, cheaper, cost effective, takes hundreds of years. So 1 in 1,000 molecule, the percentage is very less, 0.1, success rate is very poor. So based on this problem, there were a lot of Lipinski rule was derived, where he gave some hint on how to alter these physiochemical properties, where you can find some success. So the modern challenge lies from the biology, from the ADME, then comes to the synthetic problem. Because with the advancement of chemistry synthesis, whatever molecules we are able to do through a process technique, or we have integrated medicinal chemist, biochemist, as well as a process chemist. So the chemistry is not a problem now. So we go back to the biology, the target ADME profile. Once it is getting succeed in those two stages, we go for the laboratory synthesis. Okay. So these are the preclinical and clinical changes. In short, I will say, like, when you don't know about the pathophysiology. Okay. But with the modern advancement of technique, like even when we faced COVID with the lot of computational tools, bioinformatic tools, we are able to even reverse vaccine technology. Using that, we are able to produce vaccine at a faster rate. Okay. So initially, the vaccine generally takes like 10 to like at least 15 to 18 years to reach the market. And Ebola virus vaccine was the one where they used computational techniques, which was launched in the market with less than 6 to 8 years, and then the COVID vaccine. So with the advancement of a lot of computational technology, bioinformatics tools, structural biology, the speed of drug discovery process has been accelerated multiple times. Okay, but still we face the problem like pathophysiology, which we cannot address, and the limitation of animal models. What animals models? Whether the animal models exactly replicates the disease condition with respect to human behavior, and then the heterogeneity of the patient populations, and whether we do have a valid diagnostic and therapeutic markers. So these are some of the preliminary, like from the preclinical stages, where as a medicinal chemistry, we face problem because the molecule may work at one population, but it may not work at the other population. So the dose fixing, everything comes into factor. So here we work more onto the ADME type of properties. Am I audible?
Yes, sir. Audible, sir.
Okay, thank you. So this you can deep where we lack the deep understanding of pathophysiologically conditions of many disease states. The in vivo studies are favored over the in vitro studies because the researchers observe the impact on the living organisms as a whole rather than the isolated cells. So getting results in the isolated cells are generally easy, but getting the in vivo result is always a tough one. Okay. So the limitations of animal models also, like we need to develop new in vitro techniques where the three-dimensional cell growth will give you more accurate results than the two-dimensional model, where the replicating the complex microenvironment is really tough, and that is very difficult, which is one of the biggest barrier which we fail in the animal model also. Then the patients, heterogeneity of the patient populations, advances in the clinical phenotyping and genotyping also are trying to help or address these challenges in the heterogeneity of the patients, what we talk about. Then the availability of validated diagnostic therapeutic markers. So I'm rushing through these slides because these are very basics. So we get into the actual topic. Okay. So these are some of the few points which, like glimpse I've shown for which troubles as a medicinal chemistry in the preclinical and clinical studies. Now moving into the drug discovery challenges, we generally work at the interface between the biological space and the chemical space. Okay. So if you see the biological space with the protein structure, we have 10 to the power 4 to 5 with the advancement of extracrystallography, chiroelectron microscopy. So a lot of protein structures are being identified, and then they have been validated. So here we work on a lot of chemical compounds, 10 to the power of 20, where drug-like molecules are almost extensive chemical research. Whatever scaffold you have, it is reported in the chemical abstract service with its hundreds and thousands of derivatives. So we have this database available compounds. So we need to bridge the gap, narrow down the gap between the biological space and chemical space. And here, this is where we work generally as a medicinal chemist. So we try to address the gap. We have molecules, drug-like properties, or drug-like behavior, where we drug repurposing after the evolvement of the COVID, where we started screening almost one therapeutic drug molecule to the other therapeutic. The advantage of doing all this drug repurposing is you don't have to do any of the ADME studies or toxicological studies, because these drugs are already approved drugs. Unless until they show some therapeutic benefit, it can be used for the other therapeutic activity. So I'll give a glimpse on like 2-3 minutes on each slide, and then I'll go directly into the scaffold hopping and then the metabolism guided drug design, and the artificial intelligence study. So these are various techniques where a medicinal chemistry can work in the drug design aspect. So the first one, as everyone is aware, being a faculty, like these are nothing but the combinatorial library synthesis, in which you involve a great number of molecules. A combinatorial library, a chemical synthesis, where you produce hundreds of molecules. Suppose you have different reagents like A1, A2, A3, you have the other component B1, B2, B3. So by doing a lot of permutation combinations, we can produce 9 different types of compounds. We are least bothered about the therapeutic benefit at this time, because we are increased the number of molecules. So when you have more number of molecules, when you have more number of data set, when you screen it widely through HTS, whether we get a good lead molecule or poor molecules, this will try to help us getting the structure activity relationship. So from there, a computational chemist can start their his or her own investigation. So to start, we need to have some lab work done. So this is generally done by combinatorial chemistry, still one of the successful method, but not very frequent. Though they design a lot of molecules before going into the laboratory synthesis, these molecules also will be screened initially on a lot of online filters, ADME, enzyme inhibitions, and toxicological profile before getting into them. So this we called as a what you call as a building block molecules, where upon which you can build hundreds and thousands of screening compound molecules. The next one is a very familiar and easy one for the medicinal chemist, isosteric, the simple concept of isosteric replacement, where we try to replace an atom with some other atom which produces similar chemical environment. And once you get a good biological response, we call that isosteres. Giving similar biological response, we call that as the bioisosteres. So this is also a very old method, traditional method of drug design. Okay, so the objective of this isosteric replacement will be identification and development of new molecule with similar biological property to that of the parent molecule. So you change one component. So into that, you have a lot of examples like classical, non-classical isosteres, ring open, ring equivalent isosteres, all are the textbook definitions which we can use. So here it's a simple example like aminopyrin, which is an analgesic anti-inflammatory. It has got some side effect like carcinogenic. So by doing some simple isosteric replacement with an atom, you replace with a carbon atom, where you have an isopropyl type of structures, and then look for the biological activity. Of course, when you change a small hydrogen atom from this structure to this structure, 100% the biological activity is going to be disturbed. Either it will be improved, or it will be optimum, or it will be decreased. Okay, so once you look into the biological activity, again we'll investigate for the ADME properties. The next one is the de novo design, which generally creates the chemical entities based on the information of the biological target receptor or its active site, or the ligand which bounds into the active site. So from there, we try to build a model, and then we try to do a superimposable or similarity search with the inhibitor, what we observe from the library database, and then we start with the laboratory synthesis of those molecules. So these are very well-renowned molecules. So now, with the advancement of technology, we have seen like the three-dimensional structure of receptors are generally like available through the X-ray crystallography, NMR spectroscopy, or even electron microscope. And with the advancement of homology modeling also, you can acquire suitable structural information of the protein, the log. From there, you can convert the design, or start with the design molecules. The next one is the molecular hybridization approach. As the name indicates, you identify two potent pharmacophore, or the fragment of a drug molecules, which is responsible for biological activity. So it's like a synergistic effect. We know this is one is active, so 1 + 1 is going to be 2. So that is how the molecular hybridization approach is done. So if you see, like these are some potential inhibitors of breast cancer cell lines, MCF7, where you have oxodiazole, triazole. So these two 1, 3, 4 triatomic heterocycles are coupled with an open amide type of linkage. So this is molecule. We know this also comes after thorough investigation of individual fragment what is the contribution of these fragments in the biological activity, how it is involved with the binding of the receptor. Because if you touch this component which is necessary, you may completely lose the activity. So we try to retain those two potential component or pharmacophoric moiety so that we get an optimum activity. Okay. So next is the scaffold hopping. So it's a central task of medicinal chemists, generally requiring a multitude of techniques. So we have primary, secondary, tertiary, and quaternary types of scaffold hopping. I'll be talking about this in detail. So I'll move to the next other techniques, general methods of drug design. So next one is nothing but the analogs and prodrug design. The name indicates, I'm not going to talk in detail about this. So where within the same homologous series or analogs are produced, for example, if this is a carbavir, we try to produce an abacavir, which is a analog type of series where the pyrimidinone 4-one is converted into a cyclopropyl amine structure within a homologous series, and try to find out the best molecule with optimum biological activity. Apart from that, also we can, there has been some examples in the trans-diethylstilbestrol as a analog modification of estradiol, which can even produce better estrogenic activity than the original molecule. The prodrug approach varies, depends upon where you want to convert the prodrug into a drug molecule. That you design based upon the target identification. What is your target? Whether you want to pass it through the enzymes present in the GIT tract, or what is your requirements. So this depends, prodrug drug depends, generally that is applicable for the NSAID drugs, which is generally used this prodrug approach, where it can overcome the gastric irritation by masking the carboxylic acid function groups. And I'll be talking more on to the metabolism guided drug design. So this, the scaffold hopping metabolism guided drug design, I'll talk in detail in coming slides once I finish the other 3-4 small techniques, what we call it as. The other important techniques what we work on is generally the other medicinal chemists who can work is on the biology oriented synthesis. So the biological oriented synthesis is nothing but the structural simplification of the natural product scaffold. So you do a structural simplification of a natural product so that you obtain new compound class which covers biological activity space. Okay, so if you see this is an yogimbine, which is an indole type of alkaloid. You break down to the scaffold, again break down and identify. So from here you can make a lot of structural diversities, like you can make variation at this R position, R2 and R1. And then you can produce hundreds of library molecules which has got similar or close resemblance to the natural product. That is what we call it as a biology oriented synthesis. Okay. The next one is what we call it as a diversity oriented synthesis. Okay. So this diversity oriented synthesis has a different molecular scaffold possessing a high structural complexity or develop. If you see in the simple understanding, if you see the morpholine, see the morpholine is having a 2 substitution at this position. Here it is linked to 2 aromatic structure. Here they are made like a ring annulation. Here they are converted into a spiroketotype. Here in the gefitinib, it is as a part of fragment where it improves the ADME properties. Okay, so the term diversity refers to 4 principal components generally we talk about. One is the binding block or the building block diversity, a variation in the basic structural moieties around the basic common scaffold. Next one is going to be the functional group diversities, where the variation will be in the functional group present onto that. So the diversity oriented synthesis will generally have 4 principles. One will be the building block diversity. Next one is going to be the functional group diversity. And third one is the stereochemical diversity. That depends on in what way the molecule has to be oriented so that it interacts with the macromolecules. And the fourth one is the scaffold or the skeletons of many distinct molecular activity. So this will help us in designing more number of diversified molecules. So you have a lot of techniques. You can refer this particular paper, which has mentioned beautifully about the diversity-oriented synthesis. So each and every slide I've mentioned the papers of the source document, which will give a complete extensive review on the biology-oriented synthesis or diversity-oriented synthesis. These are some of the methods which helps in the diversity-oriented synthesis. Next one, I'll put a couple of minutes onto the diverted total synthesis. So this is a chemical synthesis strategy aiming of synthesizing an organic molecule of natural product analog rather than the natural product itself. So here, even if you identify the intermediate, from the intermediate also you can take a lot of diversified structures, and this is not a natural product analog, but rather than a structure which is similar to the natural product synthesis. So this approach towards the preparation of natural product like compounds, libraries by the derivation of versatile chemical intermediates also. So you can find a complete review on the diverted total synthesis in the tetrahydrolators and the Ajax paper in 2016, where they produced Professor Steve have given a complete extensive study on the diversity total synthesis. And now coming to the most easiest part, or where the current medicinal chemistry try to work, is the online computer-like design software technologies, where we use ligand-based and structure-based drug design, into which you can work on the de novo drug design also. So ligand-based is nothing but where you know the molecular structures. The basis of this will be generally from the QSAR. So when you do a QSAR, you need to have a large set of database. So that comes only when you do a lot of combinatorial library synthesis. Without having a wide range of database, it was difficult to do a QSAR. So the limitation of QSAR in those days was it has to come to the similar homologous series. But now the era has completely changed, where you have machine learning tools, where you have similarity search, a lot of online algorithms. Those algorithms require basically wide range of structural diversity. So QSAR, when you do, you want to work on indole-quinole nucleus, you are supposed to use only the same basic nucleus, but you can have structural diversity, so various number of substitutions. But with the advancement in machine learning, it completely needs wide range of diversity to understand what is the key atom or orientation required for the biological response. Next is the well-known structure-based drug design, where you understand the protein structure, do flexible or rigid docking, try to identify the active site, whether the inhibitor binds to the allosteric center or catalytic center, or it is within the active site. From there, you can design, you can understand, you can make a library of molecules, and then do drug likeness property screening, and then go for the synthesis or wet lab work. And now with this advancement, the third one is the artificial intelligence, which is an advanced version of QSAR. I put it in the simple terms like the machine learning tools. And now it has further got into deep learning methodology. So it's artificial intelligence is nothing but it is a replication of human intelligence in the machine form. It generally requires very customized hardware as well as software to develop and train a lot of algorithms using programs like Python or Java. So a lot of methods are there, like random forest, nearest neighbor, support vector, naive Bayes, consequences approaches. It all depends on how do you find your data set, how do you segregate, how do you group your molecule, how do you train the test and validate, and then that will give you a meaningful understanding of you exploring this algorithms. So I'll come back to this slide, how we perform a similarity searching before I get into the scaffold hopping. So this technique as medicinal chemistry is one of the good approach, where the molecule, when it fails at the hepatic clearance level, or when it fails at the distribution log D, or when the T half is disturbed. So there also medicinal chemistry has got a very important role to make small changes of the structural parameters, which changes its physiochemical properties, and ultimately there will be change in the biological response. So to start with, what is scaffold hopping? It's a general method to design drug by altering or replacement of a central core atoms, okay, of a known, you need to have a bioactive lead molecule. So by changing or altering or replacement the central core atom, you can reach to a target molecule which can have a similar core or completely different core. That's the advantage of scaffold hopping. If suppose you are working on one particular nucleus like indole or quinole, by doing a scaffold hopping, you can completely replace that basic scaffold with some other scaffold, where you can do again a lot of library synthesis of molecules and do an extensive screening of those studies. So they generally, scaffold hopping is applied to generate, analyze, and compare co-structure active compounds and its analog substitutions. So what is the need to do a scaffold hopping? So we try to do, as I told you, you get a good molecule, whether the compound's potency is reduced or the compound is good and potent, but they fail in the ADME-like properties. There you can use the scaffold hopping approaches. So the need comes when there is, you want to change or increase the binding affinity of the drug molecule, you make some chemical manipulation, or when the drug needs to be improved in their ADME properties to have a drug-like properties, you can do the scaffold hopping. So I'll come back to this, what is called as a maximum common structure or scaffold tree, MCS in short, we call it as. So before getting into scaffold hopping, we categorize this into 4 different types. There was a recent review on scaffold hopping. When you get time, you can please read on that. I have mentioned somewhere in my PPT, where it has completely given a thorough articles on what is a ring opening with a classical successful stories of scaffold hopping at the primary, secondary, tertiary, and 4 types of scaffold hopping. The first one I'll be talking about is the primary scaffold hopping. Okay, this primary scaffold hopping is a very preliminary, in which depends on the change of degree of change associated with the original molecules. Okay, like minor modification in the parent molecule, where you do a chemical swap of any atoms, like carbon can be replaced with any heteroatom within the backbone. You replace carbon, nitrogen, or oxygen, or a sulfur atom, small. That's why we put that into the primary scaffold hopping. So, for example, if you see here, this is nothing but your one of the drug, which is an inverse agonist on the cannabinoid B1 receptor. Okay, this drug molecule. Okay, so this was an anti-obesity drug, but failed to get approved from the US FDA because of the safety concerns. So the company AstraZeneca started working on this, where they used some of the software. There are a lot of softwares. I'll be telling then and there when required. You have CATs, you have keywords, we have Roach, we have Morph. These all helps in identifying or replacing one structure with other for the scaffold hopping structure. So in this case, when AstraZeneca was working on this CB1 receptor, the software was employed was MORPH. It's a useful tool in generating a novel aromatic ring systematically without changing the coordinates of the ring system. The coordinates will be the same. So the ring system at the core, if you see here, this is a it's a imidazole. Sorry, it's a pyrazole. And this is replaced with a thiazole. And then it is replaced with pyrrole, and then the ring expansion is done. If you see the periphery, they are not systems. There are softwares which can help you, like Morph can generate new molecules by altering individual rings to fuse the ring system, whereas they maintain the defined internal coordinates. So another molecular design approach also was used, like Morph and Roche is also based upon the scaffold hopping library generations. Okay, so by doing this, they were able to overcome the toxicity produced by this structure by replacing. However, there was a small change in the biological activity. So when you do this small change, first we look for the biological activity. When the activity is close to this one, then it will be screened for the other parameters like ADME and toxicity. So this was nothing but a typical example of primary scaffold hopping. The other examples you can see, like they are diaryl heterocycle, like sulfonide type of structures, where the thiol was replaced with imidazole, and then the hello. Okay, so the primary scaffold hopping in these 2 cases is a good, simple strategy to improve the success rate in drug discovery. Okay. And this is one more classical example where the basic porphyrin analogs were replaced with the NH was replaced with halogen, and with other atoms also like oxygen, sulfur, selenium. So it gives a lot of structural diversities. You know, like when when you do an extensive combinatorial library synthesis of one molecule by doing a scaffold hopping, primary scaffold hopping, if you're not able to get a lead molecule from that scaffold, it gives another option where you can completely get into the different scaffold, and then you can start again doing a lot of molecules, which can lead to a better hits or a lead molecule. So as I told you, primary scaffold molecule will be a simple replacement at the backbone is like a swapping of a carbon with any of the heteroatom. Next, when we move into the secondary scaffold hopping, it is more extensive, which can be like either it can open the ring or close the ring. So it manipulates. What it does is it gives a lot of flexibility to the molecule by controlling the total number of rotational free bonds, flexibility, membrane penetration, as well as the absorption characters of your drug molecules. So what happens is you can get a lot of pseudo ring structure, like you can have more drug-like structures, so that ring opening, closing, two immediate strategies to create novel scaffolds, which will give or increase flexibility of the molecules, contributes greatly not only to the inotropic components, but also membrane penetrations can also be improved. See, this is actually a classical example of O-alkoxy structures, which is an prostaglandin EP1 receptor, where there is an intramolecular hydrogen bonding. So this was one of the molecule. So by doing a scaffold hopping, or when they did a extensive study on this molecule, they tried to do a ring closing structure. You see here, there's a intramolecular hydrogen bonding between the alkoxy and the secondary amine groups. So they did a small secondary scaffold hopping and then close the structure, so which gave structural rigidity to the molecule. This ring closure design successfully locked the molecule into a bioactive conformation. If you see here, when it is ring opening, it can give rise to different conformers. By doing a ring closing structure at this particular point, we can bring the restriction of rotation, or you can bring the molecule into a one bioactive conformations. Okay. But however, the biological activity also will be checked into these molecules for secondary scaffold hopping. These are a few other examples where you have the nitrogen, where you have a ring closed structure, diatomic structures, which will be open and then further derivatized, or you have the pyrimidine molecules broken down, and other frequently ring closer open reaction, generally converting an alkyl chain to an cyclohexane, piperine, or piperidine, converting an orthohydroxy type of orthohydroxy benzyl groups into a quinazoline type of compounds. So you can screen all these molecules, look into the proper hypothesis, provided the proper orientation, you get the best biologically active conformations. So this is one more example where they lock the molecules like isoindolene type of molecules in the kinase inhibitor structure activity relation. You see here, they close to this restriction of rotation, and then it gives rise to complete a good, potent, stable, bioactive conformer. The third part comes like the replacement of a peptide bone with non-peptide moieties, which is also a very good strategy to develop or design molecules which will mimic the structural features of peptides. Because peptides, we know they are biologically unstable, and because of the imbalances of the biologically active endogenous peptide, it leads to diseased conditions, for example, like diabetes as well as cancers. So biologically active endogenous peptides, such as peptides, hormones, neuropeptides, play a vital role in the biological function of human body. So imbalance of this is always going to lead to any of the disease conditions. So the process of peptides into clinical usage is largely hampered because of the poor stability, metabolic stability, and low bioavailability of the peptide molecules. So by doing a scaffold hopping, we can design small molecules which mimic the structural features of the peptides using active conformations as template, showing a promising and challenging targets, and can overcome the stability factors as well as the good absorption parameters. These applications of the tertiary scaffold hopping can be involved in protein-protein interactions, small molecules which can mimic and interact with the protein structure. So the like some targets after using here also you have a lot of softwares like Record, I told like Morph, MORPH, and then the CATs I'll be explaining, then Record, CAVETs. These are some useful drug design scaffold tools which helps to replace a molecule into a peptide or peptidomimic groups. So I try to identify, basically it's nothing but something like pharmacophore modeling, which is given by the chemical computing group Accelerate as well as Schrodinger, which helps in designing of a peptidomimic type of drug molecules. And then coming to the fourth type of scaffold hopping, which is called as the topology-based scaffold hopping, which is more or less like the virtual screening, where the whole molecule is identified, focused on discovering of a wide range of molecules. So another application is that when new chemotype is significantly different from its templates, so the scientists might consider the process as the virtual screening rather than the scaffold hopping. Both are interlinked. Okay. The scaffold hopping focuses more on the discovering of novel core, usually ignoring the potential conflicts between the side chain and targets as a whole molecule. The complete new chemical backbone that only retains the interactions. Okay. So if you see the scaffold hopping, I'll take a classical example. Yeah. So for example, if you see some novel antibiotics, when they found like these are some antibiotics which were found to have resistance strain. So team worked on the template where they identifying some weak hits possessing potential toxicity, and then the use of shape-based rapid overlap of chemical structures, where they did a lot of superimposable scaffolding similarity search using the ROCs or OCS, rapid overlap of chemical compound structures, resulted in a two-dimensional structure towards the identification of these type of scaffolds. Other softwares also are there which will help in identifying this point-to-point distance, plane-to-angle distance. So when I talk about scaffold hopping or similarity search, how does it generally work is I'll come back to this like CAT software, which I took it from the textbook Scaffold Hopping in Medicinal Chemistry by Hugo Kimini and R.M. Manhold. So when you input the molecule, initially what happens is when you do a lot of chemical structures, the structure will be read in forms of mole file, SD files, Smiles notation. The structural fragment features will be converted into some numericals. And you can do a lot of permutation combination, correlating that numerical structure with biological activity, and then get back and identify what is the key element fragment responsible for the biological activity. So when we use like CAT's descriptors, what it does is it breaks down the molecule and gets down into the basic rings, like basic graph scaffold, what we call this as. From graph scaffold, it will try to identify, like for example, if you look into the structure, you have an aromatic ring. So that is mentioned like R, aromatic, aromatic, aromatic. You have like lipophilic structures like CS2, which are lipophilic, like that is mentioned like L. Then you have an hydrogen bond acceptor, so it is written as an acceptor, donor. So this is how the system classifies, breaks down the molecule into different fragments, and they will run n number of combinations, combination of donor plus aromatic donor plus lipophilic acceptors, and then this is how the chemical matrix system. So from that, I'll try to identify the three fragments, and then from that basic scaffold, it will be given some ring system which will be superimposed from the existing database of molecules. So if this is an example, topologically, how it is written in the form of SEINS descriptor. So this is your basic carbon scaffold. This is your whole molecule. Breakdowns into Murko's assembly where the substitution is done, the basic molecule. And from here, it brings down to the only the carbon scaffold where all the heteroatom is removed. Then where in the carbon scaffold, you will always have a saturation as well as unsaturation ring system. Then that will break down into the graph scaffold where there is only the saturated sp3 hybridized carbon structure. And that will be again reduced into the ring scaffold and reduced ring system. So this is what is generally done in the pharmacophore modeling, or you can sometimes call it as tertiary scaffold hopping. So when you identify this bond distance, bond angle, and develop a pharmacophore model into this 3-ring system, we apply this developed model into an existing database, natural product database, Campbell or molecules or zinc database, and try to find any structure with a lot of diversity matching into this ring, this bond angle and bond distance. So that is how it gives rise to a completely new chemotype by using scaffold hopping or by using virtual screening. So this is how the molecule is generally represented. When you take an example of lapotinib, how it is written like maximum common structure. So when you have hundreds and tens of molecules together, initially we'll try to find out what is the maximum common structure among those molecules. When you see like 2 molecules like this, okay, so if you look closely, these fragments, the fragments in red color are the maximum common structure. So from maximum common structure, you try to build your molecular framework. This is the one. So remove all the substitution. From there, you break down the molecule and come to the hetero framework, where the unsaturation is removed. From then you come to the graph framework, where you have only carbons into the ring structure. From there, the pharmacophore model will be generated. Okay. And now this is another method like keywords, what we call it as CAVET. Like this article was good in the Drug Discovery Today in 2010. It was a pretty old paper, like 10 years back. So the keyword generally defines, the software defines the geometry of the molecules, which generally in terms of the vectors, which is along the chemical bonds or the central core, and uses the orientation of the different vectors and angles. For example, if you look into this molecule, if this is a query molecule, see the vector, the directional vector. Like for example, if you look into R1 and R2, so the scaffold is built. So the bond angle, bond distance between this R1 and R2 is considered, and that is put into the database as a fragment. From there, it tries to identify a lot of molecules. Okay, so the keyword is one of the good software which helps in identifying the pharmacophore feature and shapes of the central scaffold which needs to be replaced. So when you have this scaffold, when I am working on these 2 directional vector, when it is matching, I can find some other completely new chemotype, but having the optimum requirement of my vectorial definition, which can have its biological activity. This is other CAT's descriptor methods, like as I told you previously, like this is nothing but how it recognizes. We as a human being looks molecule like carbon, hydrogen, nitrogen, anything. But when you look into the biological system, the macromembrane, it doesn't identify like carbon, hydrogen, oxygen, nitrogen, nothing. It will be like a magnet and an ion. The enzyme is having some electrostatic potential. It will have a positive, negative. So when the small molecule is coming, they will have some sort of affinity, electrostatic potential, binding, some force, like a magnet getting attracted towards an ion. So that is recognized in terms of the hydrogen bond donor, acceptor, positively ionizing group, charges like positive, negative. So when you have opposite charge from the protein as well as the ligand, they get affiniated or they get attracted towards each other. And once they bind, there will be a structural change. So once the structure of a protein changes, the biological function of the protein change. That is how you activate or do an agonist activity of a given enzyme. So the molecule, the descriptors will recognize them like this only. It's a lipophilic fragment, it's a donor, it's a positively ionizable because you have an NH, so which can be converted into NH plus, and then you have a carboxylic acid, which can be COO minus ion group. Okay, so this is another typical example where you can, this is the last example. I'll talk about scaffold hopping, then I will get into metabolism guided drug design. You can see, like as I told you, the vectorial direction using caveats. So if you see, this is like camphotericin, natural product, and look into the atorvastatin. So here I define the directional vector. Like if you see this 3 structure, the structure in red color is nothing but the camphotericin. So I'm defining like X1, X2, and X3 directional vector. That is my key requirement. I can put it into the database search engine by defining only these 3 parameters. So the molecular similarity approach will be done. There are a number of methods doing that. So when it is fitting into that, it exactly fits into the atorvastatin, where you have like X1, X2, and X3 type of substitution. So this was one a beautiful paper came in Natural Product Communication, but it was pretty old in 2009. So now we come to the end of one part, like scaffold hopping. So to design a scaffold hopping, you need to have a very good understanding of basic medicinal chemistry. You don't need a very sophisticated instrument, or there are a lot of free academic available online software which you can use to predict ADME, to do a secondary or tertiary scaffold hopping, or to do a flapping of this backbone, or other factors. Only thing is you need to understand the basic requirement, the fragments, the essential which needs to be retained. And you need to know when you take a scaffold, what is the problem with the scaffold? Yeah, I have good activity. It is failing in ADME. Which part? You need to have a thorough investigation where it is failing. Then you try to do a chemical manipulation. Okay. With this, I'll move on to the metabolism-guided drug design. And this is one interesting topic where medicinal chemists can really and extensively work on the metabolism, because we know like the elimination of xenobiotes often depends on converting an lipophilic molecule into a hydrophilic molecule, a biotransformation generally done in liver. And I'll be talking more on the cytochrome P450 metabolized drugs, or targeting that, because only 90% of the drugs is done by transformation only in that pathway. Okay, so any drug taken into the body doesn't accept. You don't accept any newcomer, the body doesn't accept. So the enzyme goes via either a phase 1 or phase 2. In phase 1, they can do n number of organic transformation. What we can do in a synthetic laboratory, the enzymes are able to do hydrolysis, reduction, oxidation, epoxidation, ring open, aromatic hydroxylase, any number of chemical manipulation. The enzymes are very smart to do that in phase 1, so that they can increase the hydrophilicity of the molecule and throw it extensively out of the human body. In phase 2, they're very smart. They try to do some conjugation, add some polar component, make or increase its hydrophilicity, and throw down in the form of glucuronide conjugation, sulfonation, methylation, conjugation, glutathione, so that in any phase 1 or phase 2, they don't like this new molecule getting into the body. They do chemical transformation and throw it out of the body. So when they do a chemical manipulation, either in phase 1 or phase 2, the enzyme identified some soft spots, what we call it is in the given structure, where they can introduce some chemical functional groups. Adding a simple OH into the aromatic para position makes the molecule more polar. We know like the drug molecule to remain in the human body should always have an optimum hydrophilic lipophilic balance. So when you give a higher lipophilic molecules or slightly towards lipophilic molecule, the enzyme does all this phase 1, phase 2, and then makes the molecule more hydrophilic, increases hydrophilic, and tries to excrete it from the human body. Thereby the molecules is not available in the human body to elicit its biological functions. So drug metabolism research plays an increasing role in identification of the drug metabolite with optimum balance of absorption, distribution, metabolism, excretion, as well as the toxicity characterizations. So the pharmacokinetic characterizes mainly, and this slide is very like familiar for everyone in the metabolism guided drug design, how it impacts the pharmacokinetics and pharmacodynamic parameters. So we'll be talking more on to the first pass metabolism, which is done by the liver, portal vein, gut wall, intestine, stomach, where you have a lot of cytochrome P450, 34 A, where it does a lot of phase 1 conjugation and then try to get it eliminated from the body. There are other ways by which you can overcome this by systemic circulation, IV dosing. I'm not going to address all those issues. We'll be talking once the molecule is failed at the metabolism, then how we can address those. So I'll be so this was a beautiful review paper again on metabolism guided drug design on bioorganic medicinal chemistry. So we always talk about cytochrome 450, which you have hydrophobic clusters like polar residues above the prosthetic hip. So they do a lot of organic chemical transformation like oxidation, dehydrogenation, reduction, addition, dehydration, and then hydration, hydrolysis. So anything they want, they do to get the molecule out of the body. So the strategies which can be adopted, or like we have primary, secondary, tertiary, and quaternary, 4 types of scaffolding. Here we can use 4 types of techniques to address the metabolism pathway. One is like I'll be telling in the subsequent slide, like, yeah. So the strategies to mitigate cytochrome P450 mediated metabolism is reduce the interaction within the active site. How to reduce the interaction? Try to lower the molecular weight, log P value, and add or decrease the number of hydrogen bond acceptor donor. Then try to introduce a fluorine atom, which is more polarizable at the metabolic soft sites, which is identified by the enzyme. Block the site of metabolism. If the drug is, doing metabolism at particular soft corners of the soft part of the drug molecules, we can try to introduce an electron withdrawing groups or introduce a sterically bulky group so that that particular carbon center or the reactive center is not accessible to the enzyme. Then try to modify the site of metabolism by doing either you can use a isosteric replacement, and sometimes even nowadays the drug design is overcome by replacing the hydrogen isotope with the deuterium isotope. So in general, these are the tactics we can generally use to overcome the drug metabolism. So I am talking more on the practical aspects which will not be available in many of the textbooks. So that is where my focus as a medicinal chemistry, you take a molecule where it has failed in ADME and then try to rework, identify its absorption is poor, or find out whether it is giving toxic metabolite, how we can overcome those toxic metabolite. So in short, you can reduce the lipophilicity, modulate the steric factors, introduce electronic factors, you can try to bring down the restriction of rotation by introducing conformational constraints, identify sharp spots. So for each and everything, we know, like we have worked on this metabolism guided drug design as well as scaffold hopping. So I'll talk in detail about our recent works, how did we overcome this problem. So the first one is try to reduce lipophilicity. So before getting into the lipophilicity, I need to reduce lipophilicity. You need to understand the thorough case study. You need to have the T half-life. You need to have the distribution coefficient. You need to have the hepatic liver microsome studies already reported by someone. So we take a molecule where people have failed and put it at the ADME level and gone. Try to identify. The other methods like modify the metabolism, incorporation of fluorine molecule, I'll be talking only on these 4 parameters. And then incorporation of a deuterium and a heavier isotope. If you see this example, which was reported in Bioorganic Medicinal Chemistry in 2009, it is. So this is nothing but N-aryl sulfonamide type of molecules, okay, which suffered extensive oxidation on the cyclohexyl moiety. If you see this position, this is cyclohexyl moiety. It went complete oxidation on this by the cytochrome P450 enzyme. So you can find down the IC50 value, hepatic liver microsome values, and the log T value. So when they know by complete pharmacokinetic profile, so the next group tried to rework on moving this lipophilic, lowering the lipophilic into by introducing an oxygen into a 6-membered structure, giving rise to a tetrahydropyran structure. So the lipophilicity is, see the simple structure. So however, when you make a small change, replacing a carbon with oxygen, of course, there will be a difference in the biological activity. But it's not that much bad where I have completely lost the biological activity. But see the log D and the hepatic liver clearance value. It is, yeah, good, improved, better than the previous molecule. This is how it opens up. Further, when you're able to replace the lipophilic, or when you reduce the lipophilicity by introducing oxygen atom or hydropyron, tetrahydropyron molecules, it gives an option in the next time where you can reduce again the 6-membered oxygen into a 5-membered oxygen structure. A furon type, which will always give an important opportunity. Again, you can reduce the ring size. See, the lipophilicity is completely reduced from here to here. See, the biological activity, however, it went out with 45 as the IC50 value at nanomolar concentration. See, it came down close to this, but see the improvement in the log D value distribution, and then we have a lot of variety. See, no other component was further disturbed. So small change by understanding what is the requirement of a molecule, reduce the lipophilicity, one can do the metabolism guided drug design and increase the biological activity. See here, small understanding. The other example where the lipophilicity is altered is you can have, like secondary alkyl substitutions at this position. You can look into the in vitro potency IC50 value of this molecule, and then oxetane, where the lipophilicity is reduced by introduction of oxygen atom. Further, it is converted into a tertiary butyl alcohol type. See the IC50 value, they're close in the range. Of course, in nanomolar concentration 9, it went good, and it is a bit okay, comparable. But see the T half-life, you can increase drastically from 6.0 to 76 and greater than 300. So you can do a lot of chemical manipulation. The only thing is we need to identify which particular fragment, what is the role. See the log D value distribution coefficient. So reduce the lipophilicity is one good option, provided you identify this particular feature needs to be altered in the given molecule. Here are other case examples where you can alter. So this is again, see, you need to have extensive liver metabolic stability studies reported, then you can start working on these molecules. Okay. If you see here, again, it's a piperidine type of molecule. There's two hydrogens, or in the carbon introduce oxygen, morpholine, sulfonide, and then you introduce a carboxylic acid group. See the activity at nanomolar concentration. Activity is good. There is an improved parameter pharmacokinetic, whether you check the human or mice liver hepato enzymes. So the activity is good. So this gives another option, because again you do different fragment changes in the desired biological activity. It is going to completely throw you out of the potency. You may completely get out with 100-fold increase or decrease in the activity. So you try to identify, and in this study they identify this is a problem. The molecule is more lipophilic, so that can be cut down by introducing an oxygen or another diheteroatoms, making the molecule polar molecules. So this is one where you try to identify the lipophilic molecule, try to reduce by introducing the more polar atoms, simple atoms. It can give rise to a lot of structural diversity. Next is you try to modify the site of metabolism. So this is indinavir, okay, which is generally a protease enzyme inhibitors. Okay, they generally suffer the first extensive first-pass cytochrome P3A metabolisms. So people started working in two fragments, like this part where they change this part as well as this part. So I try to change the this particular fragment with aminochronal. This fragments, this particular P2 positions, where they replace this with these two substitutions. You can look into these two structures. They introduce an oxygen atom, making the molecule slightly polar or reducing the lipophilicity of the molecule. And this was the other metabolic site where they completely redid the pyridyl moiety into furopyridine type of molecules. So which gave us. So this did not have any much biological importance, where they played only extensive role in the metabolic pathway. So these people were able to do the chemical manipulation at this position and this position, and they were able to, like a twofold increase in the T half in rats and the dog when they were doing the molecule number 13 by improving the hydrophilic parameter of this molecule. So you can modify the site of metabolism. These are other typical examples like calcium channel blockers, diltiazem, where they did extensive work. If you see here, this is nothing but you have an tertiary amine group, you have a dimethyl group, so the enzyme can do demethylation. Okay, so to overcome that, scientists has worked and kept like a secondary NH group, and then they try to cyclize this, convert into a pyrrolidine type of molecules. See here, the small change, small chemical manipulations. So that demethylation, N-methylation, O-methylation, esters, hydrolysis, do a cyclic ring closer, bring the restriction of rotation, rotatable bonds. So all this you have to identify in the given molecule. So do not start from a new molecule at a chemotherapeutic level. Take molecules, work where it has failed, a small structural change, and then you can identify the progress of the molecules. Next one is the incorporation. In many of the drug molecules where you can where you find a fluorine atom introduced into the drug molecule, in many cases it is to overcome or the difficulties faced in the metabolism processes. This tactic of introducing the fluorine atom relies on the powerful electron withdrawing effects of fluorine atom and the strength of the carbon and fluorine bond, carbon-fluorine bond, especially in the aromatic or in the carbon aliphatic structures. This bond is very strong, like when you have a carbon and fluorine, the bond strength is 108 kilocalories per mole and strongest between carbon and any other atom. Okay, this helps in identifying the soft spots, introduce the fluorine atom into that. So breaking the fluorine atom and then do a chemical manipulation by the enzyme is really very tough because the bond is very strong. And that makes the molecule also very polar in nature. So this is one typical example, which is a GPR 119 receptor antagonist and fluoroethyl groups. See the ethyl, it was a methyl group and then replaced with a fluorine atom with like see the EC50 value, the activity, it was 40, and here it is an 18 nanomolar concentration. The ED50 value is 3.9 and 3.4. So something, like to start from nowhere, by doing this, you will have an optimum biological activity close to the starting molecule. Again, you have to further optimize. So this gives a lot of option, like to take the molecule from phase 1, phase 2 to the next level. Then increasing metabolic stability through the use of a fluorine atom here. So the enzyme will do a hydroxylation on this position, okay, meta position to this. So when you introduce a fluorine atom, that spot will be masked. So that is not readily available. So you can get, you see the inhibitory concentration is good and improved half-life, like hepatic liver clearance as well as the log T value. Okay, so you can reduce the lipophilicity of the molecule, you can block the metabolic sites, and the third one, you can introduce fluorine atom into the molecules. And the fourth one is incorporation of the deuterium molecule. It's an isotope. Okay, so recently US FDA approved deuterobenzene, which is used for the treatment in, I think it was introduced in 2018 or 19. I don't remember. Yeah. So what happened is the hydrogen isotope was replaced with the deuterium. By replacing the deuterium, there is 100% change that will be improving the physiochemical property of the molecules. Because when you have a carbon hydrogen, when you replace it with carbon deuterium, the enzyme identifies the soft spots. When they want to do some hydroxylation, breaking the carbon deuterium bond takes a lot of time. Breaking a carbon hydrogen bond is easy for the enzyme, and then they can do hydroxylation reaction. So breaking the carbon deuterium bond is a bit stronger isotopy. So the drug stays in the body for a longer time. It will always have a better pharmacokinetic parameters. So the drug dose also can be reduced drastically. Okay. So we do work on the metabolism guided drug design, where we try to we took some known molecules, some natural product like curcumin, which is always having problems in the solubility issues. We try to do a deuterium exchange by doing a chemical synthesis where we try to replace the methoxy and the OH group, and then we evaluated for the activity which is equivalent to curcumin. And now we have identified or calculated the physiochemical properties in comparison with the curcumin molecule. In the same way, we did it for the metronidazole, where we worked on the metronidazole molecules, and then we try to do a metabolic, like glucuronide conjugation. We try to mask this methyl group, try to do CS3 was being converted into CD3, and then we got optimum activity, and then we did hepatic liver microsome study, which showed a better kinetic profile. But that paper is yet to be published. So these are other examples like metabolism directed synthesis oxetane, where you can find the molecules with beta secretase inhibitor activity for the Alzheimer's disease treatments. So these are other case examples. I'm not going to read. So 4 techniques by which you can do metabolism guided drug design: reduce the lipophilicity, identify the metabolic spots, try to introduce fluorine atom, and then try to do a isotope effect by replacing a hydrogen with a deuterium method. But every method requires extensive understanding of the parent or the molecule failed at the different phase. You need to have a thorough pharmacokinetic and dynamic data reported through the patent molecules or research papers or from the company understanding. So next, I'll talk about the, I'll take my other slide where I can talk about the small inputs on the machine learning algorithms, what we do for 5-10 minutes, and then I'll finish off my session. I'm not going to start from first. I'll show some glimpse of what the machine learning approaches can do. And then, because it's completely a detailed and extensive work, which will be there. So the machine learning can help you in understanding. It's nothing but a next evolution process of QSAR, what you can say. But it is not QSAR, but the next stage of drug discovery. When we have nothing from combinatorial chemistry, we moved into QSAR, then docking dynamics. Now we went to the machine learning tool. So we have a lot of artificial intelligence in every domain of the technology, like diagnostic, which plays a very important role in the artificial, how it recognizes images, diagnostic markers. But in drug discovery, especially when you talk about medicinal chemistry, we are at a very primitive stage, because what it does is the algorithm tries to identify the molecules. Even the advanced version of machine learning nowadays, we have into like taking like a deep snap, what we call it as, like when you feed computer different images, algorithms, different images, from the images fed, they'll be able to interpret and identify whether the given model image is what, whether it's a cat or rat or whatever example you can take. When we talk about three-dimensional structure of molecule, recently I saw some couple of papers in 2020, what they use a deep snap, where they took three-dimensional structure of a molecule, they took a photographs, image, three-dimensional image, and converted into the PNG file by rotating in 45 degrees, 65 degree, 90 degree with all the coordinates. And then they compared the orientation of the molecules at different angles with biological activity, and they were able to predict the molecule developed from this deep snap imaging, identifying the three-dimensional structure with all the coordinates better than the machine learning tools. It's at a very, like primitive type. So the machine learning methods generally used for predicting drug protein, discovering the drug efficacy, or find a use of existing drug molecule, which we talk about drug repurposing, safety biomolecules, and then optimum biological activity, chemical synthesis, chemical diversity or chemical library synthesis, chemical search engines, everywhere it is applicable. So I'll give you some examples which you can go through for prediction of, how I've been working on carbonic anhydrase inhibitors. So this was reported in 2021 ACS Omega. Claudia Tui-Supron is the pioneer in carbonic anhydrase inhibitors, where they used, identifying isozyme selectivity. So when we talk about isomers in terms of chemistry, there are isozymes in terms of your biology when you talk about the protein. When we talk about carbonic anhydrase, which is essential for normal physiological functions, generally we use sulfonamides as a diuretics, and in glaucoma where we use carbonic anhydrase inhibitors. So we can use carbonic anhydrase targeting for cancer cell lines because the carbonic anhydrase enzyme has got 12 different isozymes. Into that, when you look very closely, isozyme 9 and 12 are extensively, they're overexpressed in cancer cells. So we need to identify the isozyme selectivity, and that is achieved by the machine learning features. And that was successfully done by Salvatore Galati, and the paper was in ACS Omega, how they have done this particular isozyme selectivity using the prediction of target molecules. Since I'm working on carbonic anhydrase, I'm showing you some glimpse where the isozyme selectivity is, because when you don't identify the selectivity, when the hit molecule is going to bind to all the enzyme isomers, it's of no use. It's going to be toxic more than the drug molecule. It is going to shut down all the metabolic pathway required biochemical and pathophysiological conditions. So this selectivity can be brought when there is a change in the active site of the molecules using the machine learning approach. And then this is again, these are methods by which you can do like random forest, naive Bayes, nearest neighbor group approaches, where you can do project cheminformatics modeling. So predicting of binding site of receptor and ligand interaction with the active site. So androgen, this was one paper who has worked on the androgen receptor, where by identifying the binding site of the androgen receptor, they were able to use different algorithms, the random forest and then consequences approaches, and then they came out with So you know the active site, given the fragments identification, the bond distance, bond order, vector direction, distance, image, three-dimensional structure, coordinates, and then compare with biological active site, and then try to find out the basic pharmacophore mapping, and then you can explore the molecule into the different diversified structures. So these are like random forest and support vector, because we know like this artificial intelligence and then the algorithms are extensively used for a number of other techniques, but not as a medicinal chemistry in the drug discovery. How we have to use it up to us on how do we select the database, how do we segregate the database, what are the descriptors we calculate. So that comes on the deeper understanding of how do we classify. These are generally used to classify whether it is a random vector, a support vector, or a nearest neighbor group, small methods. So for this, you can read the machine learning approaches in drug design and discovery in molecules in 2010, which they have clearly explained in detail what is a support vector, mission, how a nearest neighbor group. Because when you talk about nearest neighbor group, we try to classify the database. And the data what you take has to be absolutely reliable data, reproducible. If the data what you take is not the exact replication or reliable data where you have structural diversity, then we try to segregate the molecules into training and test. And there comes the problem. There you use all these algorithms. How do we classify this using nearest neighbor group or anyone? So see, the simple example, when you have the database like this, you put this into a category into training or test. And when you have a common point in the between, whether you want to put it into this or this, that is difficult as a human being to do, but by using some of the tools, one typical example is the nearest neighbor group. So I try to understand and run a lot of algorithm utilizing all training molecules based upon the target molecules nearest neighbor group. They run and rerun, and then they classify whether to put it into a category A or category B. So this, you need to have a deeper understanding support vector. I'm not going to talk much on this. Like these are completely on the carbonic anhydrase inhibitors, where we extensively used the machine learning approach, the algorithm like nearest neighbor group and regression analysis. So with this, I like to conclude. I like to thank you for hearing me so patiently wherever you are, if you are there. So I'll take this opportunity to thank the management, the organizer, the principal, and the head of the department, so that being a faculty, I try to talk which is doable at your laboratory rather than giving an excuse to ask for a very high sophisticated software and instruments. All this you can do at your own lab, wherever you are. If you have your laptop, you have your net connection, you have your interest, your passion, your attitude towards medicinal chemistry, you can do.